Everybody. So this is our chapter 2 recorded lecture. I just want to remind you that the recorded lectures are only meant to kind of supplement your reading. They're definitely not meant to replace reading the book with the guide. The book and the guide, those are your two best tools. I just thought that it might be nice for you to have, another way of taking in the material by listening to someone talk about it, but it's definitely not a complete way of being introduced to the material. So chapter 2, again, I still have a few 6th edition slides that I'm working with because I tweaked some of them, and then I moved on to the 5th edition, which is the book that you're using. So if they look a little different, that's why. And there's some of my slides in here too. So this particular one is really, I don't know, it's kind of like a foundational chapter, water, biochemistry, and cells, and then we're going to talk about macromolecules. Really, as we're going to talk about a lot of the things in the future chapters, we need this base information. So we need to, have some of that foundational stuff. So that's really what this particular chapter is about. So the goal is to, gosh, these were a little bit out of line. The goal is for you to, know about living organisms, the molecules that make up them, and then some of the basic types of organisms, and then how organisms are arranged. So the tree of life and evolution of a population. So. Okay, so what makes something living? This is a virus. You might recognize this. It looks like the coronavirus, although it doesn't have enough detail for us to say for sure. But are viruses living? Is this a living thing? They're not. Viruses are not alive. They are not alive. And that's because they don't fulfill all the characteristics of life. And these are the 7 things. 7 things? Oh no, there's more here. Let's see. There's 9 things. There's 9 things here that a organism has to be able to do to be considered alive. And a virus is not. It's acellular. It doesn't grow. It can't move. It can replicate, but it can't reproduce. It actually makes us replicate for it. It can't respond to external environment, which is good. That means we can kill it. It can't respond. It doesn't have metabolism, which is why it needs our cells to replicate it. It can't maintain homeostasis. Homeostasis is a term that we're gonna learn. And then it can evolve. Actually, viruses can, as you know, because variants are coming out of the coronavirus. Okay, do non-living things have any of these characteristics? No, they don't. And then more comparisons about viruses, which I already did with you. So let's talk about water, okay? Because when we're talking about living things, we really need to talk about water. Water is probably the single most important molecule to life on Earth. And water is kind of a unique molecule in that it has not that it has an oxygen and 2 hydrogens, but the oxygen and the 2 hydrogens make it unique because it's what we call polar. So, you know how like when you have a magnet, like one end, like if you have 2 of them together, they kind of, they'll stick together real nice. And then if you flip them around and put the other 2 ends together, like they repel each other. That has to do with electrons, and it's not too far off from this concept. So a water molecule has an end that it's a little bit more electronegative, so it's a little more negative, and one end where the hydrogens are, they're a little more positive, and that's because the oxygen is such a big atom, it kind of sucks electrons towards it. It's a little greedy, electronegative, greedy. And so what that means is that like water molecules themselves, when they're in interacting with one another, you can kind of get that magnetic effect where the oxygen of one molecule will attract the hydrogen of another. So you get that little hydrogen bonding, that weak attraction. And then you'd have the opposite where the oxygens would repel each other. But it also makes it really good for dissolving stuff because there's a positive side and a negative side. I mean, it's not truly positive and negative, it's slightly positive and negative, but that means that, like, if you have a molecule that is going to dissolve, it's going to dissolve better in water because, for example, this is sodium chloride, this is table salt. And if you took like table salt and you poured it in a glass of water, eventually the crystals would disappear, they dissolve. Water is the solute, and I'm sorry, salt is the solute, that's what goes in, and water is the solvent, it dissolves the salt. And it does that because this little bond here breaks apart, and the chlorine kind of steals a negative charge from the sodium. And so, the chlorine, which is negative, is going to balance nice. It's going to have that magnetic attraction to the positive hydrogens, and the positive sodium is going to have that nice magnetic attraction to the negative oxygens. You don't have to know positive and negative. Nobody is going to ask you that. I'm just trying to explain it a little bit deeper, I guess, so that when you're asked like surface level questions as to why, water makes a good solvent, you can kind of understand why. If you're looking at your guide, let me flip to that page. We are talking about the things that make water, the properties of water. So another thing is, if you have something that isn't polar, so it doesn't have that slightly positive and slightly negative charge, water doesn't mix well with it, or it doesn't mix well with water. So we kind of already know oil and water don't mix. Like, that's the phrase, but that's like, if you have cooking oil and you pour it on top of water, it's going to float. That's because oil is what we call hydrophobic. It has a phobia to water. It doesn't like water. And that's because it doesn't have those slightly positive, slightly negative, so it doesn't dissolve very well. Water is really important in facilitating chemical reactions. Terms that you need to know: reactions are the things that are changed, products are what they become. Water is cohesive, so that hydrogen bonding that I talked about, that little magnetic effects, that actually kind of stabilizes water. So that's kind of nice. It's cohesive. Water is really good at moderating temperature. There's like this term in physics that I am not an expert in being able to explain, but water has a higher specific heat capacity, which means that it can take a lot of energy before the temperature raises, and so water is really good at absorbing and maintaining heat. It doesn't change very easily, I guess is the easiest way I can say that. Okay, these ones are going to be important for you to know. So we need to start really digging into that nitty gritty of some of these things called macromolecules, because they're what forms all cells. And when we're talking about living things, we need to be able to talk about nutrition, cell building components, things like that. So we're going to start with organic molecules, because macromolecules are organic. And so what does organic mean? And in biology and chemistry, organic means something very specific. It means that it has what we call a carbon hydrogen bond. Okay, so you have to have those carbon hydrogen bonds in order to be a organic molecule. But that's not it. It can't just be carbon hydrogen bonds. It can only include atoms from carbon, hydrogen, nitrogen, oxygen, phosphorus, and or sulfur. Okay, so you can't have chlorine, you can't have sodium, You can't have aluminum or iron. Those are inorganic molecules. It can only be composed of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And I remember that if you put them all together, it's chinops. Chinops. So carbon, hydrogen bonds and chinops. And that's important too, actually, this order. We'll talk about that like just slightly. But in our cells, in our macromolecules, in our bodies, the amount of carbon is the greatest, and then we have next most we have hydrogen, next most nitrogen, and so on. And so of the organic molecules, they're all going to be composed of carbon, hydrogen, but then we have a lot of nitrogen, and then relatively less phosphorus, and sulfur is the least. Just an interesting little fact. So let's practice. Is this so how do you read these molecules actually? That's important too. Let me go back. I don't know why I have a blank slide in there. So we have the carbon and the hydrogen written, but sometimes we get lazy. So this is a double bond. That's why there's two lines there, and this is a bond, this straight line. Sometimes we get lazy and we just put like a kink and we don't write the C. So for example, here, we wrote the elements or the atoms, up here. But these little kinks here, you can assume that those kinks have a carbon and hydrogens that balance them out. And so do you see anything? We have carbon hydrogen bonds. I just pointed them out, right? Do you see anything that isn't chinops? No. So this is an organic molecule. That's glucose, actually. Is this an organic molecule? It is. Yep. It's a benzene ring. You don't have to know that. I'm just pointing out. How about this one? It's got carbon hydrogen bonds. Did you spot this? Yeah, it's chlorine. That's inorganic. Ooh, this is a fancy one. No, that one. So it's got the kinks. So it's got carbon, hydrogen. So then we just look for anything else. That's organic molecule. That's what we call an amino acid. How about this one? There's carbon bonds, but is it bonded to hydrogen? No, it's bonded to two oxygens. That's carbon, two oxygen, carbon dioxygen, carbon dioxide. Carbon dioxide is not organic. How about this schmancy thing? Got these dotted lines, meaning they're going away from you, and this one coming towards you. But let's just follow what we know. It's got the kinks, right? Oops, I moved ahead. It's got the kinks. So it's got carbon hydrogen bonds, and it doesn't have any foreign guys yet. So that one's also organic. This one's actually the example that I showed you in the previous slide. This one's just drawn differently to show like this one goes forward and that one goes back, but that's still methane. So that's organic. How about this one? No, not organic, right? No carbon hydrogen bonds. It doesn't have any atoms that don't belong there, but we got to start with the carbon hydrogen. And then this this one's busy. There's a lot going on here, right? Carbon hydrogen bonds, we see the kinks. Okay. And then check CHNOP, no sulfur. So that's organic too. That's what we call a nucleotide. This is the base unit of what makes up all DNA. Okay, so I've mentioned macromolecules. Oops, clicked ahead too fast. I mentioned macromolecules, and so we're going to talk about that. So in the cell, this is a bacterial cell, but it's a representative of lots of cells. Notice that, obviously we're mostly water, right? And then we have these organic compounds, and of those organic compounds, they're split up into these four groups, and these four groups are macromolecules. So macromolecules come in four groups: carbohydrates, nucleic acid, protein, and lipids. And we're going to talk about those in order. Okay, so we're going to start with carbohydrates. Now, what I want to point out here with the macromolecules is they start out with what we call a monomer. A monomer means like mono, one, single. So that means like the base unit. So that's just the individual unit. And some of these macromolecules function as just an individual unit. That's glucose. But sometimes if you polymerize them, so you make multiples, you make strings, then you get either a different macromolecule or you get a complete macromolecule. So in this case, many monomers or a polymer of sugar, of glucose, is a starch. And then we can actually keep going and we could say, you use lots of starches to make up cell walls, but we won't go quite that far just yet. Let's talk about carbohydrates. Carbohydrates are sugars. They're the same thing. This is a fancy word. This is a scientific word. You know them as sugars. They're organic molecules, and the reason why we have sugars in our cells is mostly for energy. It's mostly, they can be used for other things like energy storage or cell wall components, but for the most part, it's going to be energy. And so you can have a monomer, which is just a single, or you can have what we call disaccharides. You could have a monosaccharide, which is one sugar with glucose, or you can have a disaccharide, which is two sugars linked together. I believe this one is sucrose, so this is table sugar. Or you can have a polysaccharide, which is like starch, like potatoes and popcorn and things like that, bread. They look different depending on who's drawing them. We like to draw them this way so that we can see it in biology, but chemists, they like to think about things in 3D. There's another way of drawing it, which is super flat. There's different types of monosaccharides. There's deoxyribose, ribose, glycerol, mannitol. And like I said, you can smoosh them together. And when you join them together by a bond, you can make a different sugar. So I mentioned sucrose is a disaccharide. Or you can have that polysaccharide, which is many, many sugars. So you can kind of see like, this is a twisting coil. So once you string them all together, they start to twist up on each other, and you get interactions between them here. They don't bond, but they kind of play together, which is why they form that shape. And this is what we call starch, this particular polysaccharide, but there are other polysaccharides. It just means many sugars, many monosaccharides makes a polysaccharide. And what are carbohydrates used for? Well, cell wall components. So things like mushrooms, yeast, they have a nice thick cell wall, and then molds also. These are all fungi, actually. All three of these have a thick cell wall made of polysaccharides, and then the exoskeletons of insects and plants are going to have polysaccharide cell walls. But yeah, these are actually all made of a polysaccharide called chitin. So the same cell wall that makes all these fungi also makes the hard shell of insects. I thought it was kind of interesting. Okay, so I've mentioned all of these things. Carbohydrates, the way we write carbohydrates is like, if you count the number of carbons and oxygens, they're equal. And then there's twice as many hydrogens to balance all that out. So it's like CH20. And so, for example, glucose would be C6H12O6. I don't know that you really need to know that, but it might help you spot them, I guess. Major source of energy, monosaccharide, disaccharide, polysaccharide, what they're used for. So we mentioned those things. Oh my, and yes, I wrote this all here too, and you can use the slide shorts to practice, but I'm not gonna reread it all to you. Let's move on to lipids. Lipids are commonly called fats, and lipids are almost all carbon and hydrogen in these really long chains. So remember how I said you can get these kinks, and that means there's a carbon with its hydrogen. So they just kind of blew it up down here to show you that there's a carbon and a hydrogen. Now, lipids can be fats. They can also be steroids like cholesterol. And then they can also be these really important lipid that it's a category all on its own. It's called a phospholipid. And so it has these same long chains, or these two long chains represented here, um, like a fat. A fat would be called like a triglyceride. This is a fatty acid chain, and it's connected to a glycerol backbone. So you get 3 for triglyceride. That's a fat. A phospholipid has 2 fatty acid chains, and instead of being attached to a glyceride head, it's attached to what we call a phosphate head. And that's really important because this part, remember, fats don't mix with water, right? But um, this guy, it likes water. So you get one side that likes water and one side that doesn't. And we're going to talk about how that's really important in making cell membranes. Okay, so fats are triglycerides. They have 3 fatty acids with a glycerol. They're really important for storing energy. That's a lot of carbon bonds that can be broken apart. Um, and then the phospholipids are really important in forming cell membranes. Steroids, I should have mentioned, you can recognize steroids because they have these rings with all these little carbons together. And you're going to know them. Cholesterol is going to be an important one, and then also hormones are steroids. Yeah, so this is another representation of those three fatty chains attached to a glycerol. It's just flipped over on the other side. Here's another representation of those really long fatty acids. Fatty acids can be called what we call saturated or unsaturated. And saturated just means like every carbon has enough hydrogens that it doesn't need to bond with another carbon to balance out. If it doesn't have enough hydrogens, it'll have to bond with another carbon, and you get this little kink, and that's what we call unsaturated. So fatty acids can be saturated or they can be unsaturated. And then there's those phospholipids again. So you got the long fatty acid chains, and you got that phosphate head. And that guy, if you see it's got like a little negative there, that means that it likes to play well with water. Remember, charged things play well with water. And that's what that means, amphipathic. I'm not sure that you need to know that term. Hydrophobic, hydrophilic. I mentioned hydrophobic before, right? Phobia, fearing, don't like water, right? Fats don't like water. And this side, philic, means love. So this one is water loving. This side loves water, and this side doesn't. Sorry about that. I don't know what it's trying to connect to. Yeah, so what that means is if you put a fat in water, or a phospholipid, I'm sorry, it'll form what we call micelles, meaning all those parts that don't like water are going to arrange themselves so that they're protected, and all the parts that do like water are going to arrange themselves so they're interacting with the water. So the little fatty parts are protected on the inside. And that's really important because that's how our cell membranes are formed. So our cell is divided from the inside of the cell and the outside of the cell. And the inside of the cell is going to have what we call aqueous. It's like a watery type substance that has all kinds of things dissolved in it. And the outside of the cell is going to be the same. So that means that the fatty acid tails, the hydrophobic tails, don't want to be on the inside of the cell or the outside of the cell. So what they do, they make a double layer. So imagine this, a layer just extends all the way around until you get a complete bubble. And the inside of the bubble is the inside of the cell, and the outside of the bubble is the outside. And these phosphate heads, they're touching the outside of the cell and the inside of the cell. And the fatty acid side chains, the hydrophobic tails, they like to be in the middle of what we call that phospholipid bilayer, 2 layers. And here's another representation of a cell membrane. So just imagine that continues, all the way around. and this would be the outside of the cell, and this would be the inside of the cell. And we have other proteins and things dissolved in there, and I'm going to talk to you about those, like a sterol, like a steroid, because that's fatty. It likes to be inside. All right. Oh, no, I don't want to show you that. Sorry. Technical difficulties. There we go. This was going to take you to a YouTube video about a concept that you don't necessarily need to know. All right, steroids. Steroids are going to be recognized because they have those rings. And, you know, sugars have rings too, but you're going to see the rings connected like side by side like this. And you're often going to see a ring that has only 5 carbons. So it looks like a little house instead of 6. These guys are important for membrane fluidity. I just kind of mentioned that. Cell signaling. So hormones is signaling. It's communication between cell to cell. So that's steroids jobs, or some steroids jobs, things like testosterone and estrogen. And then again, I have the kind of breakdown all together that I won't force you to listen to me read again. All right, proteins is the next one. Proteins are, you know, chnops, CHNO, and actually sometimes they do have P and S. But what's really neat about proteins is they're made up of individual monomers, building blocks that are called amino acids. So this is a base amino acid. It has, it's called an amino acid because it has an amine group and a carboxyl group that you don't need to know. And then it has what they call a side group here, or a variable group. And that variable group is different for each amino acid. We have like 20 of them. And they all string together with what we call a peptide bond, and you end up with a polypeptide. And if you get a big enough polypeptide, it makes a protein. And yep, so I said there's 20 of them. The fact that we have all these different variable groups, these different side groups on each amino acid, means that the big structure of the protein is going to be different. So if you have a different order of amino acids in your long string, they're going to fold up differently because those side groups are going to interact with one another differently. So like you can see these yellow ones kind of want to interact, and it's very possible that like you have some that are hydrophobic, so they want to be tucked inside the protein, and some that are hydrophilic, they want to be on the outside of the protein. And so the order of amino acids is going to determine how that protein folds up. And how that protein is folded is really important because that tells us how that protein works, what that protein's job is. And we're going to get into that a little bit more. So let's break it all the way back down again, right? Proteins are made out of amino acids. Amino acids always have this base group. And they're going to have one R group for variable group, or I forgot what the book calls it already, side group or something. And you can have all different kinds, right? 20 different kinds of variable groups. And you have amino acids, when they string together, they form a peptide bond. So this is 2, so it's a dipeptide. And eventually you get a polypeptide. And if you get a big enough polypeptide, you end up with what's called a protein. So proteins have several layers of structure. They have that primary structure, which is the order of amino acids. And then as they start to fold up, they create what we call secondary structure, and then tertiary 3 for 3D. That's the 3-dimensional structure. So that's the full structure of amino acids. So primary, which is the order of amino acids. You have a secondary structure, which is like it's starting to fold up. Like, so for example, here you have this curlicue, they call it a helix. See how this protein here, this 3-dimensional structure, the tertiary structure, has these curlicues represented in there. So that means like When I'm in class, the way I illustrate this is I take the phone cord off the telephone in class, and you know the old school phones where they have the headset and then they have that long coil cord that connects it to the base. If I took that long coil cord, that's the secondary structure, and then I wadded the cord up into my hand, into my fist, that would be the tertiary structure. So you have a wad of coils. And some proteins actually have what we call quaternary structure, which means that they're not functional unless you have many or multiple three-dimensional structures that fit together. Oh boy, that's a lot. How they're held together, remember those variable groups or those side chains? Remember the order of amino acids dictates how they fold, and that's because those variable groups bond together or interact together in some way. Okay, so this is the one where I broke it all down to you organized. And then let's move on to the last one, which is nucleic acids. Nucleic acids are made up of a monomer that has three parts. It has a sugar, it has a phosphate, and it has a nitrogenous base. And the nitrogenous bases can be slightly different. So this is a single nucleotide. This is the monomer, and this S is the sugar, the P is the phosphate, and then this is our nucleotide, or I'm sorry, our nitrogenous base. The whole thing's a nucleotide. This is the nitrogenous base. And you can have different ones of those. And what's represented here is DNA. And so there are 4 different nitrogenous bases that we can talk about, but let's look at it one more time. So you have a sugar, you have a phosphate group, and you have a nitrogenous base. Oh dear, I'm sorry. Yeah. So different nitrogenous bases that you can have. You can have guanine, adenine, cytosine, thymine, and uracil. And nucleic acids are what make up DNA in our bodies and RNA. DNA uses these 4, guanine, adenine, cytosine, and thymine. And oh dear. DNA uses guanine, adenine, cytosine, and thymine. And RNA uses, instead of thymine, it uses uracil instead. And that's how one of the ways we can tell the difference between DNA and RNA. There's a few other ways. But. So a nucleotide by itself doesn't do a lot. There's a few special nucleotides that we may talk about later, but this is supposed to say linked. I'm sorry, the word got cut off by my picture. But in order to have DNA, you need to have a polymer. And as you know, you've probably seen pictures of DNA like that very long helix structure, this one. That's many nucleotides. So remember the red is the sugar and the purple is the phosphate. And then these rungs are the nitrogenous bases that interact with one another. And so, you know, there's many there. Let me go back to the previous slide. So sugar, phosphate, and then the nitrogenous base. And you take all of these and you string them along together, and that's one side of the DNA. That's one side. And then you actually, we call it a double helix because it has this other side. DNA is made up of 2 strands of polynucleotides. RNA is actually just one strand of polynucleotide. And the sugar is different. You don't have to know this, but maybe it'll help you know one of the reasons why one's named DNA and the other's named RNA. So see if I can go back here. The sugar is called a ribose sugar. And in DNA, this oxygen's missing. So it's called deoxyribonucleic acids, right? Because it's missing the oxygen, whereas RNA is just a ribonucleic acid because it has that oxygen. So that's one more reason. So you've got the different type of sugar, slightly, right? difference of an oxygen. One is made of 2 strands, the other is made of only 1, and one uses thymine and the other uses uracil. And actually that's they bond between those. So notice you have like G bonds with C and A bonds with T. And so that's also something that we know about DNA and RNA. Here's another comparison. I like this picture. This one really sums it all up, with the exception of the sugar that you don't need to know anyway. Two strands, 1 strand, and then these are the ones that bond, or these are the nitrogenous bases, and these are the nitrogenous bases. And the difference is uracil. C bonds with G and A bonds with T. So you see here, T, A, by color coding, or in this one, G, C. Oh my, another one. Okay, I think you probably get the point, but this one's nice because it has everything all laid out very, very much for you. And there's my summary slide. So, quick little check-in with you. Which category of macromolecules includes testosterone and estrogen? What did we just learn? Lipids, right? They're sterols, and these ones are hormones, and they fall in the lipids category. Okay, just really quickly, since we're talking about cells and what makes things, what composes them and what makes them alive, we need to talk about 2 very big basic cell types, prokaryotic cells and eukaryotic cells. So all living things are made of cells. And there's 2 major types of cells, prokaryotes and eukaryotes, and they contain all those macromolecules. Like our cells are built of the same macromolecules, they're just kind of arranged slightly differently. So the difference between a prokaryote and a eukaryote, the definition of the difference between a prokaryote and a eukaryote is this right here, a membrane-bound organelle. And let's see if I can go back a slide. Oh, that wasn't helpful. It doesn't show it. So the nucleus, which is where the DNA is housed, is surrounded by a membrane in a eukaryotic cell, and in a prokaryotic cell, there is no membrane around the DNA. It's not as free floating as they make it look here. And there are other little things inside a eukaryotic cell that have a membrane around them. Remember that phospholipid bilayer. And of course, the whole cell has a phospholipid bilayer. All cells have that though. So that's the major difference, membrane-bound organelles. But then because of that, there's no true nucleus in a prokaryote. And then because there's so much more in eukaryotes, they tend to be a little bit larger. Prokaryotes are all single cellular, whereas eukaryotes can be single-celled or multiple cells. I don't know that I like this simple and complex. As a microbiologist, like I'm kind of offended on behalf of prokaryotes for that, because it's not true. And not all prokaryotes have a cell wall either, but now we're getting into my field where I'm going to be nitpicky, so we'll just move on. All right, so natural selection. So natural selection is, we're jumping all over the place here, is something that drives what we call evolution. And it basically, if a natural mutation happens in a group of organisms, and then like that mutation makes that organism more fit, is what we call it, more able to reproduce. So it survives something, or maybe it's more attractive to a mate, or maybe it can eat better. In the case of beak size and shape, which is one of the things that Darwin observed when he started coming up with this, then the population is naturally going to evolve or drift in the direction of better fitness. And wow, this is really a big leap because there's so much more involved here that I want to tell you about, but I'll try and keep it simple. So the theory of evolution, though, does kind of help us organize the what we call the tree of life. And we don't know what the common ancestor was, but we do know like that we all had a common ancestor. Like all the living things on Earth came from some common ancestor. And by following the path of evolution, by looking at those tiny changes, we can look back and we can see where we had common ancestors and where our common human common ancestors split off from others, and others split off from each other. One thing that you might think is kind of interesting, bacteria and archaea are prokaryotes, so they're the ones that don't have the membrane bound organelles, and then like everything else is a eukaryote, including us, right? We fall in the animals group. We as eukaryotes are actually more closely related to archaea than we are to bacteria. And I'm going to just let you look up archaea because they're kind of fun prokaryotes that are unique. We usually only know bacteria. Okay. All right. What cell component is found only in eukaryotic cells and not prokaryotic cells? A nucleus, right? A membrane bound organelle, a true nucleus. All right, and there is your learning outcomes. So characteristics common to living, those 9 things: value of water, cohesiveness, polarity, it's a good solvent. What makes a molecule organic? Carbon hydrogen bonds and synapse. 4 macromolecules. And that's protein, carbohydrates, nucleic acids, and lipids. And then you need to know all about them. 2 major categories of organisms: prokaryotes and eukaryotes. Membrane bound organelles versus not is the best way to separate them. The tree of life is organized based on following patterns of evolution, and natural selection, being more fit and being able to reproduce is what drives that evolution. Okay, that's the end of chapter 2.